we move from bioethics to high technology, stem cells, and Dr. Miodrag Stojkovic will present this topic. Dr. Stojkovic received his PhD in Munich, studying in vitro production of bovine embryos and trophoblastic tissue, and has a long-standing experience in both embryology and stem cell biology. Dr. Stojkovic is currently president of the Institute of Molecular Genetics and Genetic Engineering in Belgrade. Please.
So thanks a lot. Thank you for inviting me. It's a great pleasure to be back here in Spain, since I was working in Valencia 4 years. Before I was working in United Kingdom also 4 years, and before United Kingdom I was working 13 years in Germany. So finally I'm home now. Yeah? I mean in Serbia. So what I would like to introduce is, or to make some summary and overview, what we have done, what we are doing, and where we are going with the stem cell research, but also developmental biology and embryology. Just to remind you that we have basically 3 different classes of cells. You know, somatics, germ, stem cells. Somatics are almost everything we see, we have, and use: skin, muscle, blood cells, etc. Germ cells, the cells that are responsible for reproduction, propagation of the species. And of course, stem cell, the mother of all cells, or cellula madre, again, is the master cell, which gives you the rise to all different cell types in the human body. Theoretically and practically more and more every day, we see this, the possibility, 220 different cell types. So, how the somatic cells are established? Same in animals, similar in a human. You have the fertilized, totipotent fertilized oocytes, which divided and cleaved and give you blastocyst, and then in a blastocyst you have the source of inner cell mass and trophectoderm, and inner cell mass, epiblast, will give you pluripotent stem cells or embryonic stem cells, and later that will be baby, and trophectoderm cells that will be placenta. So for us it's important to know all these steps, because this is usually actually what happens, what we would like to learn, what happens in vivo, and also to put this under in vitro conditions. Because we would like to know how ectoderm, endoderm, and mesoderm develops, how they differentiate, and how we have, in the gastrula stage, how we get germ cells which give you sperm and egg. So this is the question which we would like to know, because we would like to know how the germline is established. And you have basically 2 different ways in different organisms. The one we call preformation or segregation, and one is the epigenesis or recreation. The first one is connected to some amphibians, birds, C. elegans, Drosophila, and many, many other organisms. When I say model organism, I mean something what we are learning from, also from frogs, from amphibians, from birds, because many of these signaling mechanisms and pathways are actually conserved, so also that you can apply them to mammals. Not all of them, but plenty of them. For instance, like Bruno, like Vasa, gene which is all very important for development of gonads. In mammals, epigenesis, you are aware what means epigenesis, and this is the very interesting, I will say, phenomenon, because it's seen in all mammals and has some shared features with preformation. But epigenesis is important to us because during the epigenesis, we have plenty of different mechanisms of gene silencing and gene activation. And also, what we have here, this is for mouse. From epiblast, we will get primordial germ cells. Here how this happens in a mouse. In a human, happens between 21st and 23rd day after fertilization, while the embryo is approximately 2.5 millimeters large. You see on your right side, mouse embryonic stem cells. A very similar picture is this is a human EG cells. They look completely similar. Of course, this is a mouse, this is human, but they have also similar behavior in a plastic dish, and this is the work which was pioneering work from Chamblot et al. In 1997. And these are pluripotent pluripotent stem cells. So that means they have the ability to give you ectoderm, mesoderm, in endoderm, but not the entire human body. Primordial germ cells, this is what we would like to know. They are connected to migration, and this is what we don't know how this happens. There's plenty of mechanisms and genes involved, but also the extracellular matrix is involved in migration of primordial germ cells. But in vitro primordial germ cells can differentiate into embryonal germ cells, also pluripotent cells, and this differentiation is similar to reprogramming or somatic nucleus seen in oocyte and embryonic stem cells. And embryonic stem cells are cells which are isolated from inner cell mass, and they are defined as cells which can self-renew. That means they can expand. Whether they are immortal, this is a question mark. But for instance, while we derived the first stem cell line in Newcastle in 2004, this line still behaves like we derived yesterday. So we don't see any changes on all markers, on pluripotency, karyotype, et cetera, so they're still like in 2004. And the most important for the stem cells and for stem cell biologists is you have from possibility that from stem cell have two daughter stem cells, and then the stem cell can make decision. They can give you precursor cell, and this precursor cell can give you functional mature cell. So that means this cell is lineage commitment cell, and that means they can give you adult or somatic cells, for instance, skin. So one skin and liver and cardiomyocytes and oligodendrocytes, et cetera, they all were embryonic stem cell. So therefore, for your information, we put this as a short, I would say, the list, because you are, and we all every day more and more confused what kind of cells we have, stem cells we have. We have embryonic stem cells and adult stem cells, and embryonic stem cells are embryonal stem cells, embryo germ cells, epiblast stem cells, also pluripotent and embryonic. And number 4, very interesting and very promising, induced pluripotent stem cells, or iPS cells. Somebody calls them embryonic-like stem cells, somebody calls them synthetic stem cells. I will explain you later why is this. And of course we have adult stem cells, but again, adult stem cells are all from embryo or embryonic stem cells. Embryonic stem cells were first derived in a mouse in 1981, and to derive the first line in a human, the scientists needed 18 years. In 1998, the first line was derived in the United States by Jamie Thompson. So that means we had plenty of things to learn during these 18 years, and especially about the cultural conditions and what we need to give inner cell mass, to propagate inner cell mass, and to obtain fully characterized pluripotent embryonic stem cell line. Pluripotent, that means, again, can give you germline and all our somatic tissues, can different under in vitro conditions, and this is to us very important because we would like to know the prescription, the recipe, how to drive, how to target the differentiation. The one is lesson we have to learn how to keep them in the pluripotent state, and the second lesson is how to drive or how to do targeted differentiation. Second point, also very interesting, you can genetically modify embryonic stem cells. So this means you can introduce different vectors and different genes, and you can drive this differentiation. And of course, finally, having pluripotent stem cells in a plastic dish, we were able to identify the genes which are essential for embryonic stem cell pluripotency. One is OCT4, NANOC as a gatekeeper, SOX, et cetera. So very important genes which keep pluripotent stem cells as a pluripotent stem cells. And I mentioned that we can use also and manipulate stem cells. This is our work. We use human embryonic stem cells, and we made a construct with VASA. I mentioned already VASA, why it's important for establishment and derivation of PG cells. And we made a construct with green fluorescent protein and introduced this together with VASA and transfected human embryonic stem cells, and after a while, you will see here on your right-hand side, so these are colonies of human embryonic stem cells, and these green are the primordial germ cells, as expressed here with green fluorescent protein. We've done also from FACS sorting analysis, and you see here the fraction is 0.8% of these cells are PG cells. If you do more intracellular and cellular characterization of this, then you recognize that they express typical markers for PG cells, and they're losing some markers also of pluripotency. So that means you have under in vitro condition the source of primitive cells which can give you sperm cells and oocytes. But here's the problem. The problem is that embryonic stem cells derived oocytes enter meiosis, but only enter, but they are limited in meiotic progression under in vitro conditions. So that means our biggest problem is how to obtain from PG cells mature, functional sperm and oocytes, sperm cells and oocytes. This is a huge problem for the human, and the people who are involved also in IVF clinics, because you have every day to deal with the patients, like we also do in my country with azoospermia or low level of ovarian reserve. So, of course, the science is learns always and learns from the nature. So if you do some couple of tricks, if you have embryonic stem cells derived PG cells and you put them under so-called stem cell environment or environment for these cells, for instance, in an ovary, these PG cells will give you oocytes. So that means they need something from the ovary and also from the testes to develop either to oocytes or to sperm cells. So this is where our problem begins. This part is what makes us a headache. So under in vitro conditions, you can have plenty of PG cells. Meanwhile, there are protocols that you can have 20% of the PG cells under in vitro condition. Some stem cell lines even spontaneously differentiate toward PG cells. But the major problem for us is how to get here, how to obtain oocytes or sperm under in vitro condition. So with this type of stem cells, we might be closer to solve this problem. The first problem we could solve is that to obtain patient-specific stems. Of course, if you have some patient and he has azoospermia, of course, first he will ask, could you make sperm from the stem cells? And iPS cells are induced pluripotent stem cells, and these are patient-specific stem cells. In 2005, the first work was published in Cell by Yamanaka and his co-workers, and these stem cells were derived from the skin cells. So this means from the fibroblast, and these fibroblasts were manipulated with four different genes. They were reprogrammed, and biological clock was turned from the fibroblast, from the skin cells, back in the pluripotent embryonic-like stem cell. So that means, in a lay language, that means that from all of you, we can now obtain embryonic stem cells without creating embryos. What we need is 2 square millimeters of your skin, put the fibroblast in a plastic dish, and then transfect these cells and manipulate these cells with the different genes, and then we have embryonic-like stem cells, because they behave like embryonic stem cells. They are pluripotent. They can give you the same cells as embryonic stem cells, and they look exactly the same way like embryonic stem cell too. There's some differences, but they are more similar, I would say, to embryonic stem cells, the other cells we have in our hands. So why is this important? This is important because here on this slide are iPS cells which can produce DOPA. DOPA, you know, this is necessary for some patients, like to treat some debilitating human diseases. But these iPS cells can be also used to derive sperm. This is again animal work, and this was demonstrated 2 years ago using a mice as a model. Again, the scientists from Japan, they used embryonic stem cells and iPS cells to derive sperm. So what they have done, they derived PG cells again under in vitro conditions. The second part, which is in vivo part, they used again the tissue, the testicles, and in tubulus ceniferi, they were able to obtain sperm. You have here the sperm, and then the offspring using after IVF treatment and using ICSI. So that means they started from embryonic stem cells and they started from iPS cells. That means from iPS cells from skin, and then they derived sperm at the end of this story. The same for ovaries. The same group demonstrated also that they are able to derive also the oocytes from iPS cells using, again, the small trick, and this is using a aggregation of PG cells with ovary tissue and using a reconstruction of ovaries. And you see here, these are oocytes derived from embryonic stem cells. So these oocytes are able to cleave after in vitro fertilization. These are nice blastocysts, and these are offspring derived from stem cells, which are used for derivation of oocytes. Again, the same for induced pluripotent stem cells. So that means for embryonic-like stem cells or iPS cells. Again, you see the oocytes here. Here are fertilized, cleaving embryos, and again, the plenty of healthy offspring derived after sperm injection and IVF treatment of sperm derived from iPS cells. And again, these iPS cells were derived from skin. So that means, if you see the animal model, that means it is possible from skin to obtain sperm and to obtain oocytes. So using iPS technology, that's possible. These are two scientists behind this work. They spent 10 years to develop all these protocols, including the first part, the in vitro part, to obtain PG cells, but also, now for the second part, also to use the in vivo model or to use animal model to obtain sperm and oocyte. These two scientists were worked for years in United Kingdom with a scientist actually who is I would say, the supervisor of these two, and who published last month in a Cell paper. This is a paper Iri et al. This is Azam Surami from United Kingdom, and he demonstrated that for the first time, they're able to drive differentiation also under in vitro condition using human embryonic stem cells and induce pluripotent stem cell using culture conditions and different growth factors to solve the first part, in vitro condition to obtain PG cells. So that's the first part. And of course, he identified the SOX17, who is the responsible for the signaling pathway and for driving this differentiation towards germ cells. So this is clear. This is the first part, could be repeated in a human. But unfortunately, we need to wait for the second part. The second part is, of course, the more challenging. I'm convinced that the first question will be solved very soon with all technologies we have. Gene expression profiling should be not the problem to explain the mechanism and the signaling pathways which are necessary, and also growth factors which we need to drive differentiation of iPS cells or embryonic stem cells towards PG cells and later sperm and oocytes. But the second and the third question is, we will take plenty of time to solve this problem, how to make the PG cells and to convert the PG cells to mature oocytes without transplantation, without the second part, and without in vivo work. And of course, the most important is how to repeat this mouse work in humans to produce PG cells for infertility treatment, because again, the azoospermia and the lack of the oocytes, at least what we see in our clinic is that every fifth and sixth patient has the problem with the lack of sperm or low level of oocytes. So with these three questions, I would like to thank you once again for the invitation, and I'm using the opportunity also to thank my previous and present co-workers, the previous, of course, University of Munich, University of Newcastle, and Centro de Investigación Príncipe Felipe here in Valencia, and now in human genetic of the medical faculty, and also the co-workers which we had in Cambridge in the Babrahim Institute, with a study, very nice study about the epigenetics of early human embryos and also about the differentiation of human embryonic stem cells. Thank you very much for your attention. Thanks a lot.
Thank you very much for this very interesting talk illustrating new possibilities, future possibilities for our patients. Now I would like to hear if there are some questions from a technical point of view, but also ethical point of view, like we also have here next to us our specialist in ethics about this future possibility.
Thank you for this talk, and I think is important what you were saying to clarify the in vitro gametes in human are really not here yet, and probably they will not be here for a while. And I think it's important because sometimes we have questions from patients that have heard about some stem cell therapy that somehow would help them reproductively speaking, and is sometimes tempting to say that we are almost there, but actually it seems like we are not.
Not yet, no.
But from a more of a technical point of view, considering how right now the success in the mice has always come after an in vivo passage, I was wondering if you think that this is due to the importance of the niche, because we know the spermatogonia and oogonia, they only exist really in a niche, very much so, and actually every stem cell does. And it seems that we are losing this portion in vitro. So what's your take on that?
Yes, I think you're completely right. What we are missing is the answer, what they need also under in vivo conditions. This is what we don't know. So you might be aware also there's a plenty of controversial publication also from a Jonathan Tilley group that they have found in ovary also stem cells, ovarian stem cells reserve, etc., etc. Or you have also speculation that even within a human body there are circulating embryonic-like stem cells, so-called vessels. So I think we don't have the right answer, what they will need under in vitro conditions. What we have done for all these years is trying to learn and to mimic this under in vitro condition. But the in vitro condition is always suboptimal, so we know this. And I'm not sure whether I'll give you the right answer, but I think the development, it's very close, because this breakthrough with iPS cells, which give us an excellent mechanism to study different also inherited diseases, but also certain points how to drive to these patient-specific stem cells, and also the breakthrough with the animal work. I think this combination might give you a very soon answer. I don't want to speculate now in 2 years. It might be stupid if you tell you in 2 years, but I tell you in 15 years, then I will maybe pessimistic. But we learn one. If you want to look how a human-assisted reproduction medicine progress, look at animal work. So this is this is not only about the efficiency of the protocol, but it's also about the safety, because still we're speaking about the pluripotent stem cells.
Do we have to be concerned about epigenetic? And if so, for how many generations could be transmitted? Eventual problems?
Of course, the epigenetics is an important issue because the iPS cells itself are epigenetically reprogrammed. So there's a huge, I didn't mention this, but there's some differences also in the genetic profile between iPS cells and human embryonic stem cells. Both are pluripotent, yes, but there's some differences in the genetic profile. What will happen with the genetic profile of the gametes after derivation or differentiation iPS cells, I can give you an example via this slide, which you have seen with the mouse. The plenty of oocytes, I didn't mention this, were with 3 pronuclei, abnormally fertilized. So that means there are already some mistake, okay? There's probably need also to improve this part. It is, as I had mentioned, very important to make this efficient, but also to make it safe, the whole procedure. The second issue is, which I would like to mention, is there's also the, of course, we have now the ethical issue raised whether one person can give you sperm and oocytes. If you have the iPS cells from the one person, theoretically can give you oocytes and sperm, yeah. To your information, all cells which were used, human embryonic or iPS cells to derive oocytes, were female. And for sperm were male. So that means not.
Are there other questions? So if there are no other questions, I would like to make one, maybe also to the people over here from the ethical part of view. So I guess this is a new option that you already are thinking about. And could you tell us what are the few the points that on ethically that are under debate for this eventually future possibility?
I don't know, the mic. Yeah. I think the main point right now is safety issues for the epigenetics. As said, I think some people are arguing that it would be better to go through embryonic stem cells, through somatic cell nuclear transfer. Also for the mitochondria, I understood that that might be the safer option. And also, I think a big issue is how are we ever going to test this in humans? Because as you said, now the PGCLCs, they're going through, they're retransplanted to the testes, but obviously we can't do this in humans.
No, that's the problem what I mentioned. The second part remains the problem. Yeah. That's in vivo work. So this is, we have to find the solution how to put all this in vivo under in vitro conditions. So this might take some time, yeah. And what was the first one?
Whether it would be better to go through embryonic stem cells rather than iPS cells.
But this is not patient-specific then. If you have the iPS cells from the patient who suffers of azoospermia, you have his embryonic-like stem cells. You know, so that means you can create, let's say, his sperm.
Yeah, then you'd have to go through somatic cell nuclear transfer.
Yeah, it's only iPS, but nuclear transfer is not the option. I done plenty of things in a nuclear transfer field, and there's a low efficiency. Luck, you have the logistic problem with the oocytes, and I mean, how much will cost everything is also one issue.
Yeah, that was going to be my other question. Do you think it will ever be affordable, or?
You know, we are thinking about also, when we started with human embryonic stem cell research 15 years ago, they told us no way that this will be applied one day in the clinics. The embryonic stem cells are already in a clinic, and also iPS cells in Japan already to treat some eye diseases. So that means if helps, I mean, disease is much more expensive than the treatment. It will be, yeah.
Okay,